and on understanding low-dose effects on cell organisms. He also taught NE162 in our department once. And we are working, as some of you know, also in establishing the medical physics certificate program with UCSF, with the Department of Radiation Oncology. And we hope to have Sylvain to teach one of the graduate level classes. So they we envision a 262 class. Hopefully soon, very soon. Like in the fall, it looks like fall 17 for the first time, as part of the medical physics program. So Sylvain is as a scientist at Lawrence Berkeley Lab, spending his time mainly at Potter Street, where at the formerly Life Science Division, I don't even know what the name of the name of the new division. That was just a piece of reorganization.
Biosciences, I guess.
Biosciences, so within biosciences, formerly Life Sciences, where he specializes in the study of high throughput fluorescence microscopy and DNA damage and the risk of DNA damage quantification, image analysis, and computer modeling for radiation risk. In addition to his research at LBL, he is a co-founder of Accident Biotechnology, a company using his high throughput DNA damage technology to provide phenotypic tests for individuals, research institutes, clinics, and hospitals. Okay, so I'm looking forward to your presentation.
Thank you, Kai, and thank you everyone for being here. So the work I'm going to talk about today are really the modeling aspects. So I put those two pictures here because John was those are my last two postdocs in the lab, and John worked on a lot of the NASA work we've done with modeling, and Anne-Catherine has taken over the NASA project. And so I'm going to talk a lot about modeling today, so that's their work. So before I go there, I mean, the thing that people don't, so everybody knows that this year the Nobel Prize was given for DNA repair. 3 individuals got it. And back, I think, in the 70s or 80s, the recipient of that prize mentioned that actually DNA is not a macromolecule that is a static thing that never changes. Actually, it's very much something that gets broken all the time. And it requires this amazing repair machinery to fix it all the time. And so everyone here in this room, by tomorrow, you will have every single cell in your body will have 1 to 10 DNA double-strand breaks per cell, every single cell, and you're going to repair it just fine. What you do, your lifestyle, your environment, your genetics, all these things affect how this repair takes place, and that's what triggers the sensitivity to cancer and radiation in general. So let me go over this. You have DNA damage, and you have this loop where the DNA damage gets repaired, and then you basically, as long as you have a good repair, everything is fine, you have non-cancer cells. But once in a while, you have a misrepair or unrepaired cells. Unrepaired cells typically lead to cell death. It's a mechanism that protects you from trouble. Misrepair cells is the one thing that could happen where you could escape this whole checkpoint thing and you may get a cancer cell. And so in terms of the radiation, what we know is that basically, everybody knows LNT here, linear no threshold. Is that ring familiar? Yeah. So the idea behind the regulatory process for radiation is called LNT, linear no threshold. That means that there is no such thing as safe level of radiation. Even a little bit is dangerous to you. An interesting concept, since we know that we live in a world that is radioactive, so we live in a dangerous world, I guess. Anyway, so the idea is it's really pushed by the repair process. Because when you think about DNA damage, if you double the dose, you double the number of DNA damage, you double the probability of having mutation, you double the probability of having cancer. And that's great if we are a big bag of cells, I guess it would be right, but our bag is quite complex and there's a lot of communication happening. So it's actually not so simple. Nevertheless, anything focused on DNA damage has been called targeted effect. The target is the DNA, and this is the way we've been modeling radiation for the past 50 years. Okay? The two big endpoints here is cell death for toxicity, mutation for cancer. Okay? the past 15 years, and I've been one of the person involved with this research, is what we call non-targeted effect. It's changing the entire picture. It's complicating things. Non-targeted effects are very different. They're not linear with those. They're actually non-linear. They tend to be like a switch response where you see nothing and so you see something, and it's not dose dependent. I gave you a bunch of stuff there that you can think of. The one that probably talks the most to you will be inflammation and genomic instability. We know that radiation can trigger, in some people, some chronic inflammation that doesn't come off for a long time. That's a bad thing. Why some people do it, why some other people don't do it, open question. That complicates the story. Okay? Having said that, there's also, because we're very oriented towards negative effect, because that's what we're kind of looking for, there are other phenomena that are also non-targeted effects that are beneficial to humans. It's called the hormetic response. It's such a taboo word that most of us never use it. But the literature has a lot of information about that. Animals, the most classic example is you get those animals that are prone to get cancer. It's a Japanese study. And they put those animals in a very low chronic level of radiation, and then what they discover is these animals tend to have cancer later in life. Some kind of vaccine, reaction from the radiation. So those are called hormetic response. They're very complicated and very controversial. Without going into details here, the concept of the lab is to really use experimental data, feed that information into computer models, and kind of like going into a loop where we can mimic the response, pretty much like what you guys do with Monte Carlo simulation, where you benchmark your code towards things that have been measured experimentally, tune it, go back. Except that we have a big disadvantage against you guys is that our cross-section are really terrible. And we have only a few cross-section, and the rest is pretty much a guess. So what I use is, we call them automata or agent-based model. So think about this space here. Basically, each point in this image is actually, each pixel is a cell. And I give them a way to behave. So it's pretty much a Monte Carlo simulation, but now instead of tracking particles, we're going to track pixels that have, basically they are a unit of life, cells, okay? Automata were developed by Conway in the 70s. He put an announcement on a mathematics journal where he asked people to predict the behavior of a chessboard-like things where black is live, white is dead, or something like this, and then he gives rules. And the goal was to predict the behavior of this system. They discovered that mathematically it was almost impossible to predict the emerging property of that system. So the mathematics are very poor in this level. And then we have these kind of like emerging properties. So that's what our code is. And it's really much along the line with artificial intelligence. So some of the people working with me tend to work also in machine learning, this kind of thing. So we have a strong interest here. This is the path to cancer again. This is called Mulgavkar in the 80s developed this theory. It's called the two-stage clonal expansion. It can be a multi-stage clonal expansion. The idea is that to get cancer, you need to have two mutations that are hitting specific region of the genome that are involved into either the brake system of the cells or the accelerator of the cells. The accelerator is your proliferation. So if you think of cancer as a car, well, if you think of a cell as a car, cancer would be if your accelerator is jammed, you still can use your brake. So you're accelerating, but you can still slow down the car. It's not great, but you can control it. If your brake is broken and your accelerator is jammed, you're pretty much out of luck. That's a cancer cell. So we have two checkpoints. And so basically, and proliferation is a key feature here. Every time you divide, every time you proliferate, you have an opportunity to have a mutation that can be passed on to the progeny. So this frequency mu1 and the frequency mu2s are two phenomena where here you have what we call the initiation, where the cells become abnormal. And typically what it will gain here is an advantage in proliferation. And eventually you're going to get another mutation that will lead to a transformation. That typically is getting away from all the rules of behavior. For instance, if you're a breast cell, we work on breast primarily because it's a very sensitive organ to radiation. So if the breast cells decides that it will stop listening to the environmental cues and grow outside its duct, well, you have a cancer. And typically that's the next step, transformation. You have progression, you have more mutations, there's a lot more mutation in cancer, but those are the two required conditions. And so mathematically, those you can derive the number of cancer coming up with age But we reproduce this with our automata. So we start with a healthy tissue. So it's our breast here. It's a big slab of cells, and then each pixel is a normal cells. And then eventually we have a mutation mu1 that is plugged in, and we have a cell cycle where the cells divide regularly. I'm going to go over this in a minute. And eventually we can produce, at 60 years old, let's say here we have a tumor that's coming up. So let me show you how we do it. So that's the flow chart for this. So we're zooming on those pixels. Each of those guys are cells, and then you have simple things where you may have cell death. I will go in a minute how we do the cell death thing. So whenever a cell's died, this guy has been through a Monte Carlo approach, this cell has been checked as dead. In the next cycle, the cell is dead, you have a cavity, you need to replace the cell with the new cells. And so typically the surrounding cells will replace these cells through a random, it's picked up randomly. And then whoever divide here, the new cells being produced has a probability mu1 of acquiring a mutation that is a cancer mutation. When that happens, if that's the case, now you have initiated cells. And then the next step is that if this initiated cell is next to another cell that dies through another cell death cycle, if that guy can divide again a second time, you now have the probability of getting a second mutation that will give you neoplastic cells. It's an accelerated thing. Each of these events here, this probability and this one is very rare. It's 10 to the minus 5. I'll give you the number maybe. So now what I have to do is tune the parameter to reproduce what we see in epidemiologic data. So the goal is to create those artificial human cohorts and create like epidemiologic data from a computer, not from the reality. So for the breast, what we have is that's the menstrual cycle of a woman. It's a 28-day cycle. Everything is idealized, but what's interesting is a young woman, 20-year-old, will have a very strong cycle where that's the probability of apoptosis, means cell death. So a young woman is going to have, the breast is actually very plastic at that age, and the end of the duct's got those ball called alveolar, and that's really where that's the sac where the milk is going to be. And so those things at each cycles get disrupted, destroyed, and a new one coming up. So that's where the cell death happens, not the tube, but really the end of the tubes. As you get older, this cycle is weaker, so you have less cell death, and eventually in menopause, you have no more cycle at all, and it's pretty damn flat, and very little cell death. And death is going to be the engine for cancer. So unfortunately, this machinery to give life is also an expensive machine for women, because it also creates that risk of cancer, because it's a high turnover tissue, and every time a cell dies, it has the probability of acquiring mutation. That's the model. We put the menopause at 50 here, but we have a better model now where we have a Gaussian distribution here. I'm not going to talk about that. It's not very important for this talk, but the important thing is this. We tune mu1 and mu2 so that basically you can reproduce the cancer incidence as a function of age for breast cancer. So far I've done nothing except fitting epidemiologic data. Now what's interesting is this. Let's plug in this model what we know about radiation. So radiation acts in two ways. As I mentioned, you can either induce cell death, and that's this death induced for different doses, 0.1 gray, 0.5 gray, 1.5, and so on. That's the level of cell death spread over 14 days after the exposure to radiation. If you were to get exposed, you see a wave of death that is spread over 14 days. The radiation itself also induces this mutation rate. It's a spike. You have the natural frequency of mutation, mu1 and mu2, the one I was just mentioning for spontaneous. But you also have this perturbation. Only in one day you're going to have this wave of mutation that happens, and then they will disappear, okay? Let's plug that in, and let's ask the code what happens to my artificial cohort. What we see is this. We can predict, so the gold standard for radiation risk is the ABOM data survival. Okay, so that's the data produced by Preston on breast and breast cancer observed in the Japanese population after radiation from the atomic bomb. And our prediction so that the average age was 30. So that's the average age of exposure for these people. So most women were 30 years old. Okay. This is our prediction. We are lower than what we see. Actually, the curve we see from the ABOM looks like the people were 20 years old when they got exposed. So the younger you are, the more likely you will get cancer. And it's simple. It's because you have more time to develop cancer. Okay? So interesting. We're missing something in our code. We predict pretty well, but not perfectly. That's where we're going to plug in something else. there's something else happening. And this something else is what I was mentioning earlier, the non-targeted effect. The system is responding, and there's something enhancing this response, which is this genomic instability that we observe in people. So what we're going to do now is we're going to say that radiation will induce a chronic inflammation in the tissue, but we don't know for how long this chronic inflammation is lasting, and we don't know how much there is. There's two parameters. And in fact, I'm not going to be able to resolve that. So what I have to do now is look at the excess relative risk that I just showed you here. Okay, that's excess relative risk. So that's the cancer incidence additional from the radiation in this population. And I'm going to try to match these values for two different doses. So what you do here is that if you apply with the frequency of mutation or the duration of mutation, the code generates this hyperplane. And now I know for 1 gray what is the reality from epidemiologic data. It's 0.87 is the excess relative risk. So that gives me a whole range of values that will match the experimental data. I can do the same for 3 gray. And what's interesting is that if you put those 2 points, either 1 gray or 3 gray intersection, we get the exact same curve. So now what we define here is a range of which we don't have a full conclusion, but we can explain the radiation response and the risk by saying that there is, in addition to DNA damage and cell death, toxicity, you also have this chronic inflammation. And then you can pick whatever you want. You can either say the frequency of the genomic instability is very strong but very short-lived, or you could say it's very weak but very long-lived, years. That needs to be now going, we need to go back to the experimental data to answer this question. Okay, so now I'm going to move to something else. We're going to focus on target effect, and for that, I'm going to show you what we built at LBNL. We built a mini X-ray. So it's like an X-ray beam that's 40 kilovolts. It's operated by a USB connector, so it's very weak energy. But we can deliver 2 gray per minute with that system if you get close enough. It's just a small beam. And what we do is we expose the cells on the microscope, and that's what it looks like. So that's a breast cell. We develop software that can track every single cell. So we use a nuclear stem, that's H2 bits, a histone that labels the cells. And then here we can track the cells. So we're just highlighting one cell, for instance. The beauty with this is you can give a new ID every time they divide. And then the other thing that's interesting is you can create a genealogy of when the cells got exposed to radiation, okay? And then follow the progeny. And so this is opening the door to things that could not be done before, because typically when you talk about radiation, you talk about population response. And here we are able to track individual cells and see how those 2 cells get different response. And let me show you something really fascinating. As a modeler, that really blew my mind. You see cell number 80 gave birth to 220 and 1094. And they are shown here. The label you see here is DNA damage. So we have a way to track that too. And what you see here is fascinating. 220 get exposed at the same time as 1094. They were born at the same time, so they should be identical as far as modeling is concerned. 220 get 15 damage from the radiation and decides to first divide, and then the 2 daughters come back with damage. And they themselves will repair their damage, and the new progeny will have no damage. In contrast, 1094 decides to first do the full repair, and then the next generation is clean of damage. Which tells you that basically life is extremely stochastic. I mean, within one cell division, you already have 2 cells that are completely different in their behavior. And when we did the model, we never assumed this kind of stochasticity, this kind of diversity is mind-boggling. It's actually, we're still not plugging this in the code. It's because it would create too much noise in our simulation. The thing we also notice is that if you do exposure to radiation, that's a cell before radiation, we give it 3 gray, all these little dots are DNA damage. And the things that people didn't believe us before, and now this paper is out, we show that the breaks merge together. So people was like, okay, whatever, who cares if DNA double-strand breaks merge together? Let me show you something. You can do a trick. Once we register this image, this cell, you can do a projection in time. So you take all the time frame and you collapse them into one single image. And that's what you get. So even though you get about 60 DNA double-strand breaks, so breaks in the nucleus, you can see the majority of them are located into very specific region of the nucleus, which is against the rules of physics. In physics, it should be random. There is no preferential site. But in fact, it's not the case. And that changes everything, because what we believe is happening now is that the breaks, there's basically, the nucleus would have those repair domain, and basically, when you get exposed to endogenous damage, as I told you, you have 1 to 10 breaks per cells per day. So right now, you're going to have at the most 1 or 0 breaks in each cells. And our company measures this. So I can tell you, I can confirm, we see 0.5 on average breaks per cells in the blood. Okay? So you find the breaks will go to its repair domain and get repaired. When you get exposed to 1 acute dose of radiation, you now have multiple breaks happening at the same time, and they can end up clustering into one of those domains. Now you have the probability of having 2 double-strand breaks coming together, and you can mix up the DNA. It's called translocation. And that typically can activate an oncogene, accelerator is on, or you can mess up a controller, like a tumor suppressor gene. So this is the path towards cancer. So the domain size is 1.5 micron. We also verify this with simple diffusion computations. So where we track each single DNA breaks that are moving through diffusion, we show that the square displacement is linear as a function of time, which tells you it's a diffusion process. But you can see that when you start going to very high dose, again, at high dose you see different things. High dose respond differently. We see a faster diffusion, and you also hit a constraint, which is the size of this constraint is again a domain of 1.5 microns. So we really have put our finger on something pretty interesting. There's a repair domain in there. How do you model this? So let's assume that a cell is that cylinder, and each breaks initially from the physics tells you that the breaks are randomly distributed in the nucleus. And then we tested 2 different mechanisms. We were talking to biologists, and they say you could have maybe an interaction force between the breaks through affinity. And in that case, you would have some kind of like coalescence. So we tested a coalescence model. And also, and then here the parameters that was unknown is the actual interaction range. And then we also set up a different thing, which is my favorite hypothesis, now you'll see why, which is called the repair domain, where if 3 breaks happen in the same domain, they will actually merge into one single unit for repair. And so we tested that, and based on, this is actually the number of breaks per cell as a function of dose, and you can see there is a saturation here. It's not linear. We published this in 2012. We actually used this as an argument that NT didn't make any sense. We got very criticized for that. But anyway, the beauty of this curve is now I can go back and test which parameters will fit this kind of saturation. So what happens is as you count the number of foci, those are, what we're using to detect the breaks is really an indirect measure. It's a protein that goes and binds to the repair, to the damage. And so this is a domain that actually includes 3 repair domains, 3 breaks here. So you see that the number of breaks doesn't follow linear dependency, but a saturation effect. And basically you can curve this and get the size of the repair domain. And what's interesting is the repair domain size gives us a value of 1.5. That's the way you minimize this fit here is 1.5 micron. Same number that what I just showed you with the movies. Interesting. Okay. The coalescence model also fits perfectly. So right now I can't separate either coalescence or repair domain. So now I'm going to talk about NASA work, because that's going to give us the answer to this question. So this is breast cells. We grow them in, we do those huge dish where we hit them with radiation, and they come in. So we don't do it here. We go to Brookhaven National Lab, and we go to the accelerator there. And the beam comes into the cells, and they will stop at a given depth called the Bragg Peak. It's used for therapy. So actually we can visualize a Bragg Peak here. So that's 1 millimeter, 1.5 millimeter. It's a zoom on this. The blue is the nuclear staining. The red is the label for DNA damage. And if you see here, if I do a zoom on that, you can basically see the Bragg Peak. So you see all these tracks here, and they pretty much stop here. There's a bit of the things you see at the end here is called fragmentation. It's when the particle hits an atom, head-to-head collision, and breaks the atom into 2 pieces or multiple pieces. But the majority of the breaks of the tracks stops here. That's the same breast cells visualized with the repair domain. You can see very nicely how a cosmic radiation traverses a bunch of cells at the same time. We have software that analyze this. We can extract the tracks, and we can look at the damage along the tracks and off the track. And then the physics here is something you don't learn here in the nuclear department. So when I was doing my PhD, this whole thing I had to learn it outside the department because we don't cover this topic. But basically, high LET radiation has a very different behavior, and the majority of half of the energy is deposited through what we call the core. And this is, if you were to compute if you look at this image here, the radius of this track is about 0.5 micron. The energy deposited in here by 1 GeV per nucleon iron particle is 26 gray. In contrast, outside this, we have those delta rays. This is the knockoff of the electrons that walk out. And those delta rays only deposit 0.17 gray outside. So you have that very extreme regimen where you have very high dose along the tracks, and off the track, you have very low dose. And yeah?
How's the same true for a photon if the mean free path is significantly larger? And
No.
it's not going to interact on the same cell, like if it's really close. It seems like it wouldn't be as correlated as this.
Yeah, the photon, if you were to look at it, it's pretty much uniform. There's so many tracks for regular doses that you know, at 1 gray, you're going to have basically 10 to the 4 photons in that cell. In contrast, here we have one particle per cell. It's one event. And that generates a bunch of delta rays. The delta rays will look a lot more sporadic and pretty nicely uniform. So, but along the tracks, you have a very high dose. That's why it's very different. It's a good question. So let's talk about the LET. So that LET here is 150 keV per micron. And so again, that's the visual of the Bragg Peak here. You can see how the damage drops significantly at the Bragg Peak, which is what you expect. And what you can see, if you look at the number of breaks per unit length along those tracks, and you compare it to the LET, it follows an increase, but at 150 keV per micron, it stops. And people in Germany at GSI, which are the, so I'm sorry, I forgot to mention, this theory here, the Bragg Peak theory, has been developed by the American, and actually by Berkeley, at the Bevetron, that doesn't exist anymore. So we were the pioneer. And there was a German group, and a Japanese group that was also in training back then. And the German and Japanese took this home, and now they are the pioneers, they are the leader in the world for this technology. And the Japanese claim that they have the best treatment right now for cancer, because their treatment is just, you go to Shiba, and you get great treatment there, because you see the difference between this and photon is, if you were to look at the dose profile as a function of depth, photon looks like that. It's a terrible way to treat cancer because basically you get most of the energy deposited at the surface. That's why they invented the gantry. The gantry, if you have a tumor in someone, you can shoot with different angle and you minimize the dose to the surface, but you get the tumor all the time. In contrast, the high LET looks like that. And that depth here is a function of the energy, so you can tune that. So now if you can do that, plus have a gantry, which is now what we can do, that was very difficult because you can imagine putting an accelerator on a gantry is more difficult than what we do with photons. But they can do it now. So the German and the Japanese are the leader on this, and they're using this technology. And that's what it is. Once you hit the Bragg Peak, you lose all your particles are gone.
Excuse me, Sylvain, this is protons?
Proton is the same, but proton tends to scatter more. It's not as well behaved. So the best they use is carbon.
Okay, fine.
Carbon is the best.
I understand.
But you need more juice for the carbon. So
Right.
to build a carbon therapy facility, you need $300 million.
But when you get up to iron, the Bragg Peak is not as sharp.
Oh, I think the higher the LET, the sharper it is. But as you go to lower LET, like protons,
Okay.
the lower the Z, the worse it gets. Because you can imagine you have this monster. The proton, the iron is extremely.
I thought it was the other way around, but I'm not sure.
The proton gets scattered it's light, so
Yeah.
it tends to scatter more.
Right.
That's actually the type of. So I did some simple dosimetry. NASA is going to work with me on changing this code to a more sophisticated code. Right now I use the core and penumbra, where basically the dose is constant in the core, and then you have a 1/r² dependence. But in a nutshell, Now using the same code I just showed you, I'm now back to what I did before. I'm in predictive mode, and I'm going to try to predict with my code what is the LET dependence of those damage per unit time using the theory of DNA repair domain or interaction model. And what you can see here, that's the points are the experimental data, and that's the code. And we're in prediction here. What it tells you is that the best model is this one, which is a domain model. So basically, the best fit seems to suggest that a cell by default has about 10 to 15 domains. And whenever you have multiple breaks in that domain, it will put them together and repair them together. It's like you're driving and you have an accident. They're not going to fix your car on the highway. They're going to take the car to a garage and fix it. The problem is when you have radiation, you've got 20 breaks, 20 accidents at a time. They all end up in the same shop. And then you can maybe get confused by taking the wrong parts to the wrong car. So in a nutshell, that's what our model tells you. And that changes everything because this is something fascinating. I know I'm going very fast, but just in terms of the whole message here, one of the gold standard in toxicity of radiation is what we call the survival curve. It looks like this. And you basically, with the same model, we can basically infer this kind of clustering phenomena as a lethal event. So that's new. So we're bringing these ideas that in the past, when you were to predict cell death from radiation, we use this model called the target model. And the idea is like DNA double strand breaks, those events are lethal events. And so the probability of death is 1 minus exponential minus gamma n, n being the number of breaks. What we said in this work is like, let's separate the kind of breaks. If you have a break that is isolated, it will have a certain value for lethality. But when you have a cluster, the lethality is much stronger with a parameter that we're going to fit. You can fit the data with X-ray response, and we can fit nicely. And then again, using the exact same trick, we fit with X-ray, we predict with the geometry of high LET radiation. And what you can see here is that with this model, we can predict that the high LET, the cosmic rays, are more toxic than the gamma rays. And we call this RBE, relative biological effectiveness. If you take NE162, you'll hear about it a lot. It's the quality factor that we use in health physics. That tells you that you're 1.74 times more efficient than with gamma for the same dose to induce this event, death. And actually we did further, and the NASA really liked that. They actually just funded us to do this work again with now real human tissue. Basically we predicted very nicely the LET dependence that is known from the 80s in the Beavertron. So this is very interesting how we can now predict the RBE as a function of LET for any particles, which means that this is an instrument that you can use for predicting risk. Okay, I'm going to finish on radiotherapy. I don't know how much time I have left.
15 minutes.
Okay. So, the idea is that radiation will kill cells, and the cells that are cycling are more sensitive to radiation. Okay. Now, let me show you something that is really simple, but you all know that when you get radiotherapy, you get multiple treatment, right? So the classic treatment is 2 gray every day for 5 days, and then you do that for 5 weeks, okay? So now if you look at a survival curve, we just went over this, it looks like that. Percent survival as a function of dose. And then you can see that the curve not being linear, if you give 2 times the dose, so that's a dose D, and you do 2 doses in 2 different days, the idea for radiotherapy is that you repeat the same survival curve twice. So that curve is just shifted down and you repeat it. And that gives you the survival of the tumor at this level. If you had given 2D in one shot, that's the death you would have gotten. Much more efficient. So in a nutshell, the ideal treatment is 1 single dose. And you don't need as much radiation. So why do you think we don't do that? Why are we bothering ourselves with 5 weeks treatment, 2 grays a day?
Well, that's enormous.
To reduce toxicity, right? Remember the angle thing? This is changing. And unfortunately, the US are not in this, but the Europeans are very much in favor of what we call hypofractionation. We're going to reduce the number of tumors. So And so here, that's the answer to the question I just asked. We limit it by the toxicity to the normal tissue. There's another factor that also limits the radiotherapy is the radioresistance of the tumor cells. This is what a classic protocol looks like. You get fraction every day, 5 weeks, and this is the classic curve. We call that probability of what's called the tumor control probability, TCP, and basically the more fraction, F is the number of fraction. If you do a lot of fraction with very low dose, you need a very high dose to get 50% control in the population. If you now increase the dose per fraction and reduce the number of fraction, you start seeing that curve shifting to the left, okay? Okay, it's pretty simple. So the hypofractionation is the current protocol the French are trying to push. So I went to France for 3 months, and we're setting up the code that I'm going to show you right now is actually based on the work we've done over there. They have a great training facility. And the idea is to do 2-week treatment instead of 5-week treatment with a much higher dose. Here is the problem. You talk to a doctor, and they have the protocol. It's empirical. Medicine is very much an empirical science. And a doctor knows that if they give 2 gray every day for 5 weeks, they're going to get a 75% success rate. Now you say, I want to change your protocol, and this protocol is like, what if I get 60%? I just kill 15% of my population. They won't do it. So that's a problem. So the idea is that we're going to create a code. They're going to reproduce everything they do first, show it works. Then we're going to ask the code to predict other protocols that are better, but without risking the life of people. So that's the goal here. So other big advantage. So why the French, why the Europeans are so much into this, and why the US are not? Because we have a different healthcare system. In this country, we wait to be sick to be fixed. The Europeans are much more on the prevention side. And the reason is they have a healthcare system that is a social system. It's costing them a lot of money to treat someone. They would much rather have someone not sick. So for that reason, and then the last thing is because they're on budget, it's much more economical to treat someone for a week and then go to the next patient than to treat someone for 5 weeks. On the patient side, it's also a win. Prevention is good, but even in the treatment side, hypofractionation is a better approach because those people have to find a room. They typically have, so when I was at the training center, they had people coming with RVs, and they had a whole RV park for the patient because that's one of the most economical way to get treated. They rent an RV for 5-6 weeks. They park the RV at the hospital, so they have an RV site. And I was thinking, well, why are there so many RVs? Like, well, those are all the cancer patients. They sleep here. And because radiotherapy is not something where you need to stay in the hospital. You go, you get zapped, you get out. So then where do you go? If you go in the center that's 200 miles from where you live, well, you stay at the hotel, it costs a fortune. And that's not reimbursed by the Social Security over there. They reimburse everything, but not the hotel. So for those reasons alone, you can see the advantage of hypofractionation. So that's we're setting up an international lab, and if you're interested, we're looking for a student who wants to go to France, and we're doing this exchange, so we set that up. We have funding for this. So people in my lab actually are flying to France once a year. So little incentive here. You do have to work when you go to France, though. And you go to Nantes, which is where it's the French Brittany. It's a very nice place. So anyway, if you like crepes, it's a good place to go. And François is my counterpart, is the biologist leading the group over there. So we got funding to do this. And that's the work we've done this summer. So I use the same type of code, agent-based model. I'm not going to go in detail. Now the yellow cells are the healthy tissue. This is a tumor growing into a tissue that's open. And then what we do is we create like different things. We have a map for the oxygen level. So what you see here in this is the tumor is the red and the green cells are tumor cells. The green are hypoxic tumor, and the red cells are normal tumors. That means the hypoxic cells are able to grow in low level of oxygen. So they tend to go far from those blue things, which are the blood vessel crossing the tumor. Okay, I'm going to tell you in a minute how I did the oxygen delivery in the tumor, but a tumor needs oxygen to grow. And then what we did is we looked at parameters that would make the tumor grow at the same speed of what's been published in the literature. So this is a model of prostate cancer. So they take the prostate cancer from human, they put them in mice, they actually put them, it's called orthotopic tumor. So they put them back in the prostate of the animal, and it grows as a tumor inside the prostate of the male. Okay, and so those are the experimental data for the growth of those tumors in the tissue, and the curve here is the model that we created that matches very nicely the growth kinetics. So far I'm just tuning the parameters to match reality. Okay. For the oxygen level, I use a simple tricks. I use a Gaussian filter, an imaging filter. So a vessel is like 1 pixel. So I forgot to mention something here. Things are not to scales in those models, and it doesn't really matter. The tumor is single point, they're about 15 micron wide. Those blue points are really much bigger than blood vessels. They're not cells, and so that's probably more like 100 micron wide. Okay? But for the model, it works just fine. And so the way we distribute the oxygen from those blood vessels is we went to the literature and look at the oxygen diffusion from the walls. Apply a Gaussian filter. So a Gaussian filter in imaging is pretty much a diffusion process. And we feed it the sigma of the Gaussian so that we match the diffusion levels. So that's shown here. And then now with that, we could reproduce the, there's another phenomenon that I need to talk about today. So when a blood vessel gets hit by radiation, it becomes more leaky. And so more oxygen goes through the blood vessels. So we model this with what we call the diffusion process, the perfusion process. So, and we had to fit the amount of damage you get from radiation to fit the experimental data. So that's one thing we looked at. And so what you see in the treatment, the points here are real data, and this is the simulation. As you do fraction every day, what they observe is that the level of hypoxia in the tumor drops. You have more and more oxygen in the tumor, which is a good thing, because oxygen helps killing the tumor. I don't know if you know about this. There's drugs you can take that will actually give a better uptake of oxygen in the tumor, and you get better control of the tumor. So, in effect, the fact of the blood vessels having more perfusion helps better the treatment. But now we can model it very precisely with this model. Okay. So that was another thing we put in the code. The other thing we put in the code is if you hit too hard with the radiation, you start killing the blood vessel. But you need to go with an acute dose of 7 gray to start seeing some vessel death. Okay? So all these are experimental data. We plug that in the code, and now we can reproduce those tumor control probability. So what you see here is that basically now we're in pure prediction mode. We looked at the total dose given with different protocols. So here is an example of 3 gray every day. And you can see we get a tumor control of about 30 gray. Okay? So now this is a tool that we hope the radiation oncologists can use to optimize the treatment and test different protocol. So that's kind of the kind of profile you see with this. And then so right now we're generating a bunch of different protocols, and of course the higher the dose, the less dose you need. You see you get about 18. The egg gray gives you about at 16 gray you already get 50% control. So you can really treat very fast with this system. So the final goal is this: is what we're trying to create in France right now is a computer model that will be able to predict the response of a treatment as we do the radiotherapy. So currently, what do you think is the best way to see if your treatment is working in radiotherapy? It's really simple. Think about it. If you have cancer, they start treating you. What do you think they do to know if it's working?
Don't they have biomarkers in the blood?
That'd be nice. Yeah, for prostate, I think they look at PSA level. No, they use imaging, and they see if the tumor shrinks. The problem with that, it's a terrible method. You get the answer 6 months after the treatment. So basically, you know if it didn't work after you treat the person. So it's pretty much useless. And so the medical physics world right now they're pretty desperate because Varian and all these companies, they optimize their machine so well now. They're delivering the dose so well, and they're like, we have 2 problems. One, we don't know how to sensitize the tumor better, so there's a whole field that's opening called radiosensitizer. So putting chemicals like a chemical that would be activated with radiation is one of the things they're looking at. So that's one thing. And the other thing is they're looking for ways to know if the treatment is working as they're treating the patient, not after. That doesn't exist at the moment. So when you go on a treatment, you have no idea if it's working. It takes months. And then eventually they start seeing the tumor shrink when it was already dead. But you don't know until much later. So the goal here is to create a code that will optimize the treatment and also read from the biomarker, like you just said, to predict toxicity, to predict the tumor response, and change the protocol on the fly. So there is one place right now in the US where they're doing this, not for radiotherapy, it's with prostate cancer. It's some of my colleagues there, it's MOFIT, and they managed to convince doctors to be off. They're basically treating people with computer, and the doctors have been taken out of the equation. There was a lot of resistance with this, obviously. So it took them 3 years to get the clinical trial approved, but the idea is very simple. It's those people have prostate cancer, and at the end, if you try everything and you're still dying from prostate cancer, there's a last treatment they do. It'll last 6 months, then you'll die. It's I think it's an anti-testosterone drug. It's an inhibitor, so it blocks the hormones from the tumors like these hormones to grow. So if you cut it, you start having a control of the tumor, but very quickly the tumor build up their own production of hormones. So over 6 months it adapts, and eventually the tumors grow and kills you. So what they notice is that the computer model that developed by us, they show that by using simple evolution model, you can predict, you can actually tune the level of that inhibitor and play with the biomarkers in the blood, and basically they show they can adjust the amount and keep the prostate tumors at check. So you don't get rid of them, but you keep it at check. And so in their simulation, they can show that those people can live for years under this treatment. So they convince the doctors to be off hands and to let the computer decide based on the biomarker read. And so, okay, every day, I think it's weekly, they have the panels of biomarkers, and they say, okay, that person should have an increase, that person should have a decrease, because in the field of evolution and ecosystem, it's a very advanced field, actually, and the usage of pesticides are optimized through these machines, and I think radiotherapy could also benefit from this kind of approach. So that's what we're trying to push right now. And that's pretty much it.
Okay. That's fascinating. What kind of still progress possible? Of course, in doing with the tools and technologies available, and including simulations and modeling too. Any questions? More questions?
I have plenty of questions, but I'll only have 2 for random.
Yeah.
So you're basically, what you're basically saying is that, so most of calculations that are done to calculate radiation dose and exposure and all those kinds of things assume fundamentally that there's no time dependence in that factor at all. So a lot of what you're suggesting here is that it does really matter. Like there's only so much damage these cells can take and repair fast enough.
Yeah.
So does that mean, so are you effectively pushing that sort of the tail end of this, linear no threshold model curve, or are you basically turning it concave upward?
Well,
Concave down?
yeah.
Because I know that there's a lot of people who have strong arguments about which one is actually the correct one.
So for the survival curve, they actually show that the linear chromatic model doesn't work at high
Yes.
dose. Yeah. It's actually very linear. It's interesting. So right now we're still using the alpha beta model for this, but it's something we're trying to change. But you put your finger on something more important, is the time dependency and also the adaptation. So when you go to the literature, there is no cellular work done at the moment that has described simple question. I went very fast, I apologize, but I wanted to give you more of a glimpse of what you can do with model. And this would take me really 5 or 6 hours to go in detail with this. But there's something called the growth arrest. When you hit cells with radiation, they stop dividing for a given amount of time. About 1 gray, about 14 hours, they stop because they're repairing their damage. And then they go back in. Okay, this growth arrest is well defined. In the literature, there is nothing, and we look very hard. Something describing what happens when you do a fraction. Is the cells adapting? Meaning that if I give 2 gray today, okay, I get 14 hours growth arrest. But if I do 2 gray on the same cells tomorrow, do I still get 14 hours of growth arrest? Is it less? Is it more? Is it adapting? Question and answer. And so when you start modeling this stuff, and this is really what's interesting about modeling, is it's forcing you to think through every single thing, because you got to plug it in, right? So you're like, okay, I don't have this information, where is it? So you look in the literature, you can't find it, and you call your colleagues, like, was there any publication in the 70s and the 80s? Like, oh no. So what we're doing with the French right now, we're building up a huge high throughput system that will actually go back to all these basic measurements, and they're actually going to do all these ones, which is impossible to get funded. If we were to go to NIH and say, oh, we're going back to textbook question, and we want to ask the question of growth arrest as a function of fraction, adaptation. There's no funding for this. It's like, oh, we know that. It's additive. They use this simple model. And I didn't even show you something we do with Kai that I didn't have time, but when you put those breast cells in chronic exposure with cesium, so we have a way to also use that for the live cells. We put them with radioactive cesium. If you leave them for 48 hours in cesium, and then after you shoot them with radiation, they act completely differently than if you shoot them with 1 gray without the exposure to cesium. So the chronic exposure seems to make them, they seem to repair faster from the radiation. They seem to repair better. So there's some adaptation right there. 48 hours.
Okay, second question. So this will be just the only other question I ask, which is that, do they know much about what affects the speed at which this is done? Because I know so I know there's bacterium that live inside nuclear reactors, and they are just able to repair crazy amounts of damage. Is that a function of maybe the actual length of the strands, or is it something more complicated than that?
Yeah. There's a guy who works on this. So he showed, it's fascinating. You know how bacteria dies of those high radiation? It's not through DNA damage per se, it's through protein damage. The doses are so high that it's trying to destroy the proteins. So no one ever thought of this because there's so much protein, there's so many copies of the same protein, it's never been the target for the radiation biologists. But the bacteria, so movement is a terrible thing when it comes to break, and they manage to keep their DNA very static. The spore seems to actually crystallize the DNA, so that even if you have a break in your, even if you have a break like that, the problem is if you do a double strand breaks, those things come apart. The bacteria is able to keep this structure rigid, and so after there's just an enzyme that goes and repair. So actually, the DNA can be broken like hell, and still they will repair it. But what happens is, as you hit so hard with the protein, the actual protein, we call them the key function of the cell itself are disappearing because they're losing their actual, they're losing the protein themselves, and actually they're losing the repair proteins. The repair proteins are destroyed, so they can't go repair the DNA, but DNA itself could be repaired if you have the protein. So the radioduron, the one you're talking about, they can take, 15,000 gray or something crazy?
Wow.
So
it's be a little more advanced than simple bacteria.
it's changing the field when you look at them, but we don't work on that.
Okay, yeah.
So if you get these models working very well in the several gray region, do you think you could try them in the small fraction of a gray region and see what they would produce?
Yeah, it's the low dose program. So that's harder because my feeling about the low dose regimen is I feel like the cells are actually very well suited to repair. And so I think actually you can't extrapolate back to the low dose for the same reason I was saying, right? If you have like one break at a time, it will repair just fine. So I don't think radiation is that dangerous. Having said that, the reason why LNT and the reason why they don't want to touch it too much is those non-targeted effects have some negative impact. Because, so let me tell you a story. The low dose program, I'll finish on that one. The model we have is we take the animal, the female, and we take out, when they're 3 weeks old, we take out the epithelial cells, so you just cook it out. And so you remove the nipples and the part that will branch out the tree. Those breasts they will never grow, okay? And what we do is we take breast cells from another animal, we put them in, and we take breast cells that tend to get cancer, okay? So they're primed for cancer. If you take the animal that has been removed, where you remove the breast and then you put the new breast in, okay? If you expose the animal before to radiation, so you're putting in cells in an environment that has been modified by the radiation. what we show is that two strands have completely different effect. On the biopsy, which is an animal that is prone to breast cancer, when you do that, the radiation accelerates the cancer incidence. You take the spiritus, which is a wild-type animal, it's much smaller, it jumps a lot, very resistant. That one doesn't have cancer in general. You do the same experiment. Radiation radiation actually reduces the number of cancer. So on one hand, we have and so the bottom line, that's why we created the company. I haven't talked about the company today, but our company is dedicated to identify who's at risk, and all the epidemiology should be done along with biomarkers. It's essential because right now they're taking everybody in cohort and they get those huge variations, and then you hear they expressing, oh, we need 1 million people to conclude. Fukushima, we can't conclude. Well, if you were to go back to Fukushima and you separate the people based on their sensitivity through those tests, you will get very clean data, and the error bar is going to be much smaller. But the problem is they put everybody in the same bag, so the error bars are like that, and good luck to conclude the low dose range. You can't.
Yeah. That's really, going back to there would be advanced technologies that could be extremely useful, but it's really difficult to get it funded, particularly in this country. I think the final point I wanted to ask you about the LNT, because there's strong evidence, of course, that LNT, of course, is very limited and not really sufficient to describe all these effects. And the discussion about the LNT, which is a major impact, of course, on regulation, all of that, is different in different countries. So since you mentioned France, can you maybe final comments on the different approach and to potentially overcome LNT in France, for example?
Well, I think in France, the Academy of Sciences has mentioned, but they don't believe in the LNT system. They think it's a bit of it doesn't make any sense first intuitively, because how can an entity like us that has evolved to live in a radioactive world kind of handle radiation? Almost like it's a nonsense. So that's just for that point alone, there's an argument against it. Now, in terms of the regulation, I think they depend through, I think ICRP, all these institutes are they supersede everybody. So they still use the IRR principle as low as reasonable, achievable, that stuff. Right? But I think it's difficult to comment on this because Fukushima ended up being a big mess. People have become very fearful of radiation again. And it's unfortunate because we all know that the evacuation probably killed more people than what the radiation would have done. And I think the problem is it's a political problem. It's not a scientific problem, and we need someone to really have the courage to fight it. And I don't know who that would be.
Difficult, yes. Okay.
I think Obama decided to engage in nuclear industry after 7 years.
Anyway, okay, let's thank Sylvain.